Gosman and David Dent. And the person that will be giving the presentation is David Dent.
Yep.
And he'll be talking about a very interesting technology on how to bring more nitrogen into crops. Here in Mexico, biofertilizers are a popular technology, but this is all having to do with soil interactions. And this is really something new that I hadn't heard about, where the microorganisms work actually inside the plant. So we're very much looking forward to your talk, David. Here.
Hello there. Thank you for giving up your lunchtime to come and listen to me. I hope to make it well worth your while. Feel free if there are any questions or as I go along that you don't quite understand or want clarification. Quite happy to do that for you. So yes, I'm going to be talking about the establishing of symbiotic nitrogen fixation in cereals and other non-leguminous crops. And we think this is genuinely the way forward to a greener nitrogen revolution. So who's Azotic Technologies? We're a company, but we were established in 2012 specifically to commercialise a patent from the University of Nottingham from work carried out by Professor Ted Cocking. And the bacteria that we're working with is Gluconocytobacter diazotrophicus. And I expect all of you to be able to say it fluently by the end of my presentation. Gluconocytobacter diazotrophicus. But to make life easy for everybody, including myself, we refer to it as GD. We set up in 2014 with our laboratory at BioCity, and we have 14 scientific and technical staff working on this project. So what would make an ideal nitrogen-fixing bacterium? Everybody's heard of rhizobia. Rhizobia in legumes, in root nodules, confined to the roots. This is something different. What if you actually had the ability to colonize any crop plant? What if you had the ability to move through the whole of that plant, to fix nitrogen throughout the whole of that plant? And on top of that, you could do it even in the presence of nitrogen fertilizer, so you can get an additive effect. And then if you had additional plant growth benefits, that would be an even bigger plus. And what if, of course, you were also no harm to the environment, no harm to humans or animals? This really would be an ideal microorganism for a biofertilizer. People often ask me, why have we suddenly appeared on the scene? Where has this research come from? And the term that is often used is snake oil, in the sense that nobody really believes that what we're talking about could be possible. It seems to have come out of thin air. It hasn't come out of thin air. This is a process of development of research since 1960. And the publications were in Nature originally by Professor Ted Cocking. And what he was able to do was to take a plant cell, which has got a rigid wall around it. He applied enzymes to that, naturally occurring ones from fungi, and he broke that cell wall down. So he ended up with a plant protoplast, and that allowed him to put things into that protoplast. So he put in rhizobia, because he wanted that cell to fix nitrogen. And so that's what he started doing in 1960, and he spent many, many years of his career, and he actually was made a Fellow of the Royal Society in the UK, which is the creme de la creme of scientists, because of this work. But what he was actually trying to do was put the rhizobia into a cell, then through cell tissue techniques, grow up the whole plant with the bacteria throughout the whole of the plant rather than just in root nodules, and fixing nitrogen. But he either got the bacteria into the plant fixing nitrogen, or into the cell fixing nitrogen, but then couldn't get the plant to grow, or he could get the plant to grow and the bacteria wouldn't fix nitrogen. So he failed. But he learnt so much along the way. And he learnt enough when 1988 came along and Gluconocytobacter diazotrophicus was discovered first in Brazil. And it's here in Mexico, in sugarcane. He started screening a range of these bacteria, or isolates, and found one in particular that had all the enzymes and all the characteristics he thought would make an ideal nitrogen-fixing bacteria going through the whole of the plant. And this isolate is the one that we've been working with since we set up the company in 2012. So what is it? Well, it's an obligate endophyte. that means it cannot really survive very well outside the plant. It has to be in the plant to survive, to thrive, to reproduce. But it is found in a range of high sucrose content crops. So it is in sweet potato, pineapple, you can find it, and you can also find it in sweet sorghum and mango and a number of others. There's about 15 species in total that it can be found in, but they're all high sucrose crops. So it's out there naturally, but in its natural condition it stays intercellularly. It's intercellular spaces in the high sucrose xylem vessels. It produces indole acetic acid, which is plant growth hormone, and it does produce a benefit according to that. But one of the great things about this bacteria, it doesn't have a nitrate reductase enzyme, which essentially means it doesn't register the presence of a nitrate fertilizer. And so that means it carries on doing what it does, fixing nitrogen, even if there's nitrogen fertilizer available, which is different from rhizobia, which get lazy if there's nitrogen fertilizer around. Why should they bother fixing all this nitrogen when there's another source for them to use? GD in sugarcane fixes about 60% to 80% of the plant's requirements, and it excretes about 50% of whatever it produces into the cell or into the cells surrounding it. GD has 3 enzymes, there are 2 there, there's a third as well that actually helps penetrate the cell membranes that allows this particular strain to go intracellularly, within the cells, not between the cells. And when it's in there, it forms what we call a symbiosome, and I'll explain how that works in a minute. Firstly though, intracellular colonization was actually, just over 10 years ago, quite a controversial issue. And there were lots of scientists who were very sceptical about the publication of Ted's work in 2006. They were basically saying bacteria do not go into cells, and they certainly do not fix nitrogen. The only way we know about intracellular colonization is through a root nodule. The only way we know about nitrogen fixation is within a plant is in a root nodule. So it was controversial stuff. But the root nodules, you can actually get some rhizobia that don't need root nodules. So that's not quite true. And there are other species, Gunnera for instance, with a cyanobacterium that can also go intracellular. So there are examples out there. And the more we look, we find more and more these different microorganisms that 1 are intracellular and 2 fix nitrogen within the plant. But you'll notice when we're talking about spruce or pine or palm and cactus, these are all in nutrient poor soils. So it makes sense for there to be a nitrogen fixing bacterial association with the plant. So nodules are not essential. Whatever you may be told, nodules are not essential for having nitrogen fixation in a plant from an endophyte. These 3 diagrams are quite important. The first on the left was one of the initial pictures that Ted took of a maize root tip. The black dots, any way you can see black dots in these 3 pictures, is the GD. And so it shows that the GD is actually going intracellularly. You can see some of them are on the edges and some are going inside the cells. This is a hair, single cell there of a trichome on a leaf of Lolium peren, and the black dots are a GD. But this is the one that's really important, because these here are chloroplasts. Chloroplasts, as you're all aware, take the CO2, water, energy, sunlight energy, and produce sucrose so the plant can grow. But the black dots here are the bacteria sitting alongside the chloroplasts. And the reason they do that we hypothesize they do that, that's a high sucrose content area. So they're being drawn to it because that's the energy source that they use when fixing nitrogen. So how does it all happen? Well, we apply the bacteria in a formulation to the seed. And what we're interested in doing is keeping this damn thing alive, because it doesn't like surviving in the soil at all. You can maybe even in sterile soil you can get it to live a while, but certainly in any normal soils where there are bacterial competition, it's much more difficult to keep bacteria alive. We keep it alive long enough so when the root emerges, the bacteria swims, and it swims down to this area here by the root cap. And the reason why is because this area behind the root cap is an area of undifferentiated meristematic cells. They're very simple, very basic cells. And one of the reasons we think that this bacteria can move between different crop species and colonize different crop species is because of this undifferentiated cells in this area are very simple, very basic, and common to most plant species. So it punches holes through these enzymes, through the epidermis and into the cells. And then they're taken up by endocytosis by the plant. Now at some point, and we're not sure about this at the moment, it could be that the bacteria at this point is actually seen as a pathogen. And then once it's taken up here, there must be some form of communication with the plant so that it's seen as an endosymbiont. These are things we're only just beginning to touch on. There's a wealth of information, things that we don't know, that are going to be exciting in the years ahead as we move forward. But these are all the crops we've been able to show colonization of. And you'll see this is a tremendous variation and wide range of crops. So all the cereal crops, but tea, cotton, coffee, tomato, radish, lettuce, potato, turf grass. We've even got over here oil palm with work we're doing in Malaysia. Essentially, every crop we've tried it with, we get colonization. That's not to say we get large amounts of colonization or enough to have an impact on plant growth and yield, but we haven't investigated that in all these crops. But we have invested colonization. But does it actually fix nitrogen? And this is the key. If it's going to get in there but doesn't do anything, well, what's the point? So we've looked at everything, all the different techniques that have been made available from all the rhizobial research that's gone on with root nodules and legumes, and essentially within the literature or with our own work, we have demonstrated that for every method that is used for rhizobia or demonstration of rhizobial nitrogen fixation, we have used, or somebody else has used for this species of GD. So on that basis, we've looked at the measurement of foliage greenness and chlorophyll content. In a colonized crop, it goes up. Measurement percentage of N in the biomass in a colonized crop relative to a control, it goes up. We've done labeled nitrogen studies, and I'll come back to this, NIF minus mutant studies, and the measurement nitrogenase activity in ARA. And then, of course, you have to demonstrate it in the field. We've done all this, but we've actually done a little bit more. And that makes it quite exciting, and I'll come back to it in a second. So here's our first trial that we ever did. This was back in spring of 2013. All I was interested at that time was seeing whether the formulation actually worked in a field environment and did get into the plant. We didn't expect to be getting results like this. So this is a very simple experiment in pasture grass, and I chose pasture grass because I was concerned that the bacteria wouldn't move throughout a seasonal crop or an annual crop quickly enough to have any impact. So I worked on perennial ryegrass, and we got a biomass increase, as you can see there, nearly 50% increase. Leaf nitrogen content went up and chlorophyll content went up. We were just blown away by this in the first year of trials. Now we're doing much more sophisticated stuff. And this is work not done by us, it's done by independent scientists in the university, by a PhD student with a supervisor who you'll see in a minute, Rupert Frey. And we're doing NIF minus mutant studies. Now, that all rolls off my tongue relatively easily these days. For those of you who don't know, a NIF gene is the nitrogen fixing gene. So if you create a mutant bacteria of GD that has that switched off, and then compare the wild type and the mutant, you should see differences if the nitrogen fixation is having any impact. And this is the work that was done on tomato, and we've been trying desperately to get a slide of the work we did on wheat, but we haven't managed to get into the presentation today, so my apologies for that. But what you're seeing here is, what you're seeing here is those are the two mutants, so they had the NIF genes, but they've been switched off. This is the control plant, which it has the formulation of the bacteria, but no bacteria. And this is a standard control with water. What you would expect if the NIF genes are switched off is it would be the same as the two controls, which they are. And then the wild type is producing more plant growth, larger roots, higher yield in tomatoes, and the same is true when you just work with wheat. This is the bit where we've taken it a bit further. This is really at the forefront of science, use of a new technique called a nanosims microscope. And what this very cleverly does is allow you to bombard a slide of a section and for a particular set of ions that pick out the thing that you're looking for. And if you've picked it out and it's there, it's bright red or yellowy green. If it's not there, it's blue or black. So this is just to demonstrate in culture, so this is literally from an agar plate, that we had a control, and that control is the blue bacteria there, Enterococcus facialis, and that is the GD, that is the GD, more controls around there. And then you can actually measure the amount of nitrogen by the colour. And so that one is fixing an awful lot of labelled nitrogen. That one is fixing less. And this automatically poses some questions for us to say, well, hang on, are some of these bacteria actually fixing more nitrogen than others? Is there a population with a normal distribution? We don't know yet, but this technique will allow us to test that. And we're working with Manchester University staff to actually test that hypothesis. But this is the really interesting one, because this is images taken from a colonized maize plant. So we've allowed the plant to grow, we inoculated the seed, allowed the plant to grow, and then we gave it loads of labelled nitrogen. And then we took sections, and this here is the chloroplast. And that red area there is the labelled nitrogen. So this is a colonized plant by the bacteria that's fixing nitrogen from the air, releasing it into the plant, and it's actually making its way, by whatever means we're not sure yet, but it's making its way to the chloroplast. That means you've got an incredibly efficient system, because you need the chloroplast, the chloroplast needs the nitrogen for the chlorophyll to help it photosynthesize. And if that source of nitrogen is literally sitting outside that cell wall, organelle wall, then you've got the shortest metabolic costs of all in getting the chlorophyll that's required. But that's all very nice stuff, and it's all background science, but what happens when we go to the field? So I'm sure you're all aware of normal yield response curves with nitrogen fertilizers. So as the fertilizer increases, you get to an optimum M per particular field, for a particular cultivar, for a particular crop species. So what we hypothesized is that our N-fix, our product that has the bacteria in it, what it would do at lower yields, lower N levels, it would add more nitrogen than the plant could get from the fertilizer, and you would get this curve. But we also hypothesized that you might get something different. If the bacteria is doing something else, and remember I did say it had the plant growth hormone indole acetic acid, then you might expect another response. If it's having any effect on photosynthesis, you might have another response. And don't get me wrong, this isn't so unique. People don't talk about it much, but rhizobia is known to affect photosynthesis as well. So rhizobia doesn't always just give you that response, it can give you more of these parallel lines. So just to show you what we get, this graph in fact is not one of the common ones. This is the classic nitrogen response from the field, but we don't get this usually. What we actually get are these responses. Now this is an average of 22 trials that we've carried out on maize. So this is all their yield responses, and we get these parallel lines. And you will notice that those are incredibly small error bars that we get in that. And this also then shows you, if you translate that, and so 100%, just so I make it clear, 100% is 100% of the recommended rate of fertilizer for that crop variety for that particular field. But what we get here at 0% is actually a higher percentage yield increase than we get at any of the other levels of nitrogen fertilizer. So it will add on a yield increase at 100% of the recommended rate, but you're going to get most benefits in nutrient poor soils. A lot of developing countries this could have an impact on. Wheat. We've had more trouble, more difficulty with wheat, in working with wheat. It doesn't follow the simple model that appears to work with maize. And here's an example of some of our best results, and they're a bit they're a bit mixed, and so that's the classic parallel, and that in yield increase was about 20% yield increase, which is just phenomenal. That's wheat in the UK. But sadly, we didn't get the same results the following year, but we did get the nitrogen response. Now, we don't know why we get that response one year, we get that another. It could be purely environmental conditions, but why shouldn't it be having the same impact on photosynthesis in one year than it does in another? Why shouldn't the indole acetic acid or plant growth hormones be the same between years? These are questions we just don't know yet. And then we got almost a mixed response here. So we are intrigued a great deal by wheat. Rice photosynthesis. We've looked at rice and got very good results, and I'll show you some in a minute. Part of the thinking is that we do have an impact on photosynthesis. And Eric Murchie at the University of Nottingham has been working on rice with IRI for many years. And one of the things he did, a very simple experiment, and we've now got a proper project with him, is that looking at control high end, control low end, and the biomass there is lower than you have with the nitrogen fertilizer. The end fix, there's not a lot of difference in the biomass. We weren't interested in any of that. He was going to measure what was happening in photosynthesis in the leaves. And what he's actually found is that in the treated, we're actually getting, so they've had the colonized bacteria, you're getting an increase in the maximum rate of photosynthesis. So we are getting that improvement in photosynthesis for the bacteria, perhaps because it's in association with the chloroplasts. And we're into grower validation stuff now. This is Sophie Alexander. This photograph was taken of her. She's an organic farmer in the south of the UK. The photograph was taken of her at the point at which she was holding these up and phoning us to say, I've got a difference in yield. And this was the sort of difference that she was looking at, both in terms of numbers of grains and size of grain. And you can see the greener strip going up the middle. I will show this here. Oops, sorry, that's not going to work. I wonder. Well, that isn't. No, I think I'll have to miss the video. There's a video here, and the reason I show it is a salutary lesson of what you do in a laboratory and what happens when you get out to the farmer. So we put together a formulation of the seed, which worked great in the laboratory, worked great in the field, but getting it into the field was the key. And what we actually found was that we did all the flowability tests that you do by all international standards to say these seeds will flow through a drill at the set rate. But when you put a ton of seed into a hopper, you get a completely different response. And what you actually get, or what we got at that point until we changed the formulation, was one big mass of seed. So these are very simple things that have to be tested out on the proper scale with farmers doing things the way they normally do them. And this was a salutary lesson for us, that even though we did everything by international standards, those standards didn't translate to what it meant to the farmer in the field. We started getting these yield increases, and then we got people saying, oh, well, yes, but what if the quality goes down because you've got this yield increase? Or perhaps the quality will change, so it won't be as good for milling for bread. So we started looking at quality, and this is some of 15 trials carried out in 2016. And what you see here is we're actually getting, on average, a 4% increase in protein content of the grain from the treated versus the untreated. And again, we were staggered by this, but also others were as well. Now, Hovis are a milling company in the UK who make bread and cakes and all sorts, and they looked at the grain quality, or the flour quality from the grain from the treated crops. And basically their chief scientist said he had never seen results like this in the 30 years that he'd been doing science related to milling and flour. NFIX there, for every measure they use to evaluate how good the grain is going to be for flour, NFIX was better. We then gave some to Marriage Millers, another milling company, and they looked at the bread making quality. And actually we were getting better quality of bread out of the flour that had even a lower protein content than their best. So the protein isn't just the fact it is protein, it's the type of protein that's important, and it is changing and different from what is available through nitrate fertilizers. And it's becoming an exciting story. And then we looked at corn silage with USDA as a collaborator and found we get improvement in energy content and in digestibility for treated versus untreated. And this is actually the Hovis mob doing their bread sampling and tasting of the different treatments. And so then there's potential for other crops. And why do I put this up? Well, I just want to give you a sense of, we talked about the range of crops that can be colonized, but what is the potential for yield increases? So we've been asked to look at potato. We did this just recently in Canada. The top two bars there are yield increases with NFIX relative to the controls. These are just testers, if you like, into the field. So we haven't gone down to a 0N. We've just basically said, let's have a look. Does it have any impact before we start investing a lot of resources in this? And lo and behold, it did. And we now have a major potato company who's very interested, and it's going to fund us to do research on their potato cultivars. And then everybody else has said, well, rhizobia is used in soybean, will it work with soybean? So we did some laboratory studies and we had a look at it, and we felt, yep, it colonizes, there's no problem with that, but will it have an impact on yield? And again, here we are, this was at low N, 0N, we got this big increase, and you even got a bit of an increase at the 100%. So it's not working against rhizobia in the root nodules, it's working with and producing that increase in colonization and increase in yield. This chap here is Rupert Frey at the University of Nottingham. He sent this photograph to us because he's worked in tomato over many years of his career, and he sent this photograph because he could not himself believe what he was seeing. Now, I do know, and as you all appreciate, this is in a glass house, this is under laboratory conditions, and we can all say, well, they're all perfectly ideal. But when you've only got 25% of the normal level of nitrogen in that pot, and that's untreated control, and that's the treated, you can see that's a fundamental difference in biomass that's produced, and all the measurements are associated with that in terms of crop yield in the tomatoes. And we're now trying to develop a project looking to see how quality of the tomato might change because of the presence of the bacteria. So interesting things. But none of this can happen. None of this can be a product unless the regulatory environment's right for it. Now, we all know about all the issues about GMOs. This doesn't have any of those issues at all. The regulatory environment for biofertilizers around the world is very favourable indeed. So that means we actually carry out trials in the UK without having to give any notification of doing so. It has a very low risk status. That's very important from a commercial point of view of us getting it into the marketplace. One of the things I point out, and it's particularly relevant that I point it out here, we are completely compliant with the CBD and the Nagoya Protocol on benefit sharing. And why is it important here? It's important here because the strain that we work with, the isolate of the bacteria of Glucosidobacter diazotrophicus, was isolated here in Mexico. The university that we take the patent from, we license the patent from the University of Nottingham, they have a relationship with the UNAM, and they have a benefit sharing arrangement. We don't know what that is, but it's obviously satisfies UNAM, and they're so that essentially any money that we have to give to the university is shared here in Mexico with UNAM. So we're fully compliant, and we insisted that this was the case. We weren't prepared to take to market without that being the case. So that means there could be much more research going on in the future in UNAM associated with this type of approach. So, UK, currently no registration permit required. We're registered for sale already in 20 states in the US. And this spring, we go into our pre-commercial trials with over 300 maize growers. And they'll each be probably applying about the treatments to about 25 acres. And then 2,000 and 19 will be into sales. So we're nearly there. We're a pre-revenue company, and that's always the big risk time for any company, but sales will be next year, and they'll start off in May. Right, next. So application of CFIA. The Canadians actually have the most stringent regulations with regard to biofertilizers. And so one of the things we've done, we've carried out trials in Canada already, and as you've seen from the data, we've carried out those trials under permit. But even so, they're not really worried about this product. But we're going through the regulatory process just to make sure the most stringent organisation in the world, or regulatory authority, allows this product to be used. It's just a pragmatic approach. In the UK, we're certified as being organic, which for organic growers is a good thing, and Sophie Alexander was an organic grower. In all of the culture collections around the world, it's designated as a hazard level 1, which is the lowest, and in the FDA it's seen as a grass product, generally thought of as safe, and the acetic acid bacteria are used generally in the food industry. So your vinegar comes from using a form of GD. And I actually have here, just so I can probably show you, on here, a business card from a woman who was actually working on Gluconocetabacter xylenus and producing materials from its fermentation process. And she was actually wearing clothes made out of this material. So the bacteria, whether it's in food, whether it's in textiles, whatever it's used for, it's generally considered as safe. And people have actually been eating GD for millennia. And they've been eating them in sugarcane juice, in pineapple, sweet potato, mango. You may have even eaten some GD this morning for your breakfast. So, what's the implications of all of this? Well, we started off as a company thinking we're going to be a nitrogen substitution company. So we're going to say, for all the right reasons for climate change and sustainability, we will be able to reduce fertilizer use by 25% or 50%, or if we're lucky, even more. When we started getting the yield increases, even at 100% of the recommended rate, it transformed our business. And it's transformed it so that we now have a number of areas in which we can actually have an impact. And in the UK, we have what we call nitrate vulnerable zones. 66% of agriculture in the UK is a nitrate vulnerable zone, where the use of nitrogen fertilizers are restricted. I'm talking to the UK and meet with the Minister literally the week after next, discussing the impact that this product potentially can have, and the rules that might be introduced by government to encourage farmers to use this type of product. Enhance yields across commercial agriculture, we can do that. So if food security is an issue around the world, carry on using 100% of the recommended rate of nitrogen and get an increase in yield boost. That's not what we're really about, but it would still be an option. But for commercial agriculture, reducing the amount of nitrogen fertilizers being used and reducing nitrous oxide emissions by using our bacteria as a supplement mean they can maintain yields the way they've always done with the fertilizers. But in developing countries, we're now very much aware that we can, in nitrogen poor soils, we can actually increase yields quite significantly for developing countries, smallholder farmers, and we are working basically to see how we can translate what we have for commercial agriculture into subsistence agriculture. And what that really means is that because we can get the bacteria into potato, which is a vegetative reproducing crop, then the same would apply, we believe, with cassava, with yams. And so it opens up a whole range of different crops for subsistence farmers that we have and there is no commercial interest in that, but it could make a difference to people's livelihoods in a major, major way. So I'm going to leave you there, and I'm quite happy to answer any level questions, and if there are any that I feel I can't answer, I'll just pass them over to my colleague here, Nick, and I'm sure he'll be happy to talk to you. Thank you.
Well, thank you very much, David. Well, we open the floor for questions, and please use the microphone because there are other people that are joining remotely. Nelly?
Very well done.
Hi. Could you tell us a little bit about the practicality of how seed is treated? At what time before planting should you inoculate and all of that? And if you have any idea on costs? For
Okay,
farms?
so no, that's actually quite a few questions, but I'll answer them all. So we, one of the things you do in a company is you essentially have a product development chain, and you take it through that process. So first of all, it's proof of concept, and then it's a case of taking it all the way through to eventual commercial field trials. To get to that proof of concept, you have to do an awful lot of work. And what we're doing is our first product is a liquid inoculant onto the seed. And then we have a second product that will be out a few years later, which is a more sophisticated seed coating. And the difference is that first product, that inoculant, is an excess of liquid in which you have to put your seeds. The second product means you've gone from, say, 200 mils per kilo of seed down to 20 mils per kilo of seed. And that then becomes an industrial seed treatment process. And so we're still at the inoculant stage, but what we've done is chosen crops to work with, excepting wheat, where we can actually use this inoculant. So in-furrow processes for application of fertilizers and seeds in the US for maize is ideal. It's putting on a large volume of liquid on top of the seed as it goes along the furrow. So we're doing our trials next year with the commercial growers, or this year with the commercial growers, using in-furrow. Rice is another example. You prime your rice so that it germinates before you put it in. So part of that priming process, and there are a number of places you can actually intervene, we are then using the GD inoculant in that process. So it becomes a seamless process for the farmer. And that can be done in a bucket. It really has not got to be done at any sophisticated industrial scale at all. And it just means giving that product to the farmer. But it does mean that we can actually produce it as a dried powder. And we haven't made that economical as yet, but we're working on that with people who know how to do this. And on pricing, I think the simplest thing to say is the packaging costs more than the product. So we actually have a big margin to play with in terms of delivery of this product to a wide range of different audiences. And one of the things, obviously, is the concern that from a commercial perspective, you could say, well, we'll give this product for maize, so it's going to be used in Zambia. But what's going to stop somebody in Zambia then taking that product and selling it into Europe? And the difference is in the way that you provide your packaging on a scale that's relevant to its use. No European farmer is going to be tearing up packets or taking out bottles for 1 acre or 1 hectare or half a hectare. They're dealing on a different scale. So there are ways of managing it. So we can actually carve up, if you like, the market and deliver different products at different prices for those markets. Does that answer your questions? Okay. Thank you.
You talked a lot about your colonization across species. Have you also tested how the variation is within a species? So, to different varieties?
Yes.
Yes. Yeah.
That's a very good question. And we don't see any varietal differences in maize. We see a little bit of varietal differences in rice, but that was mainly in the response to our product with germination. And so we modified the formulation to get that right now. But we get most variation in wheat and wheat cultivars. Now, that doesn't mean to say we don't get any colonization. We do get colonization across all of those that we're working with, and we're working with collections. And one of the reasons we're here is to talk about what we'd like to be able to do with the CIMMYT, certainly wheat collection. And it's a case of us. We think we're getting colonization at the level of about 10 CFUs per mil from a seedling, and that at 10 to the 4 or 10 to the 5 is really
the level that we need in the plant for it to work effectively. And we get variation in that. So sometimes you get 10 to the 1, 10 to the 2, 10 to the 3, and then you get some in 10 to 4, 10 to 5. What we're finding is in some of the older cultivars, we get higher colonization levels of the bacteria. And this just raises so many interesting things to potentially do. It could literally be a case of we've bred out the ability for the symbiotic relationship for these bacteria. And some of the indication of that is given from, I spent time in Mauritius with the sugarcane industry there, and they have lost in all of their modern cultivars the GD in their sucrose, in their sugarcane. They've bred it out. And so they're literally now saying, we want to put the GD back into our modern cultivars. So we know as a process, you can breed out the association with the bacteria. So that's one of the reasons we're here to discuss what we might be able to do on a collaborative basis to look at those sorts of issues, because they're quite exciting ones.
So my question actually goes to Yvonne. Are there plans to test this product in Mexico, and is CIMMYT involved in those plans?
Yeah, well,
I think putting you on the spot.
you know, we were talking about potential funding sources, but you know, we still need to talk a little bit later today. But I'm thinking that perhaps we can use a little bit of the money from CSIRO to test some of these products in maize and wheat at a small scale. But if we really want to do a large project, we would really need to find additional funding.
Any other questions? Yeah.
I have a question. Does the bacteria colonize the seed and then propagate with plants?
That's a very interesting question, and the answer is yes. So the bacteria will go into the developing grain, but not in every crop, and at different levels in different crops. So the transmission to another generation, from a commercial point of view, that wouldn't be a really good thing from sustainability or being able to provide this product as a public good. It probably would be a good thing. But what we've found is that you only get a very low level of colonization of the grain that then translates into a growing plant with the bacteria in it in the next generation. And to give you an example, in wheat, we've never been able to demonstrate colonization in the grain and carryover. In barley, it's about 6%. So 6% of the grains have GD in them, and then some of those will have the GD in the following generation. Oilseed rape is really interesting. 16% of the seeds in oilseed rape contain GD in the next generation. So we took the next generation again and then looked to see whether they were there. We couldn't find any GD. It's all about probabilities, because how many bacteria do you need to get into that grain to establish the colony in the next generation? How long do they survive in there? How many of those grains are going to be needed to give you a sustainable population? All sorts of issues of that kind. We're only just touching on it, but we do know that there is some level of seed transmission, and you would perhaps expect that for an obligate endophyte.
I'm an economist, so at the end of the day, I would like to know some numbers. I'm sorry if it is a stupid question, but do you have any study on this, that if this technology is applied in some crop, then how much other fertilizer, nitrogen fertilizer, I don't need to apply because of this bacteria?
Yeah.
And what's the monetary value of that one?
Yeah. So that is where we started from. We wanted to be able to cut the costs for a farmer of their nitrogen fertilizer. So there had to be a significant reduction. And what we've found over the period of our trials is that we can get, on average, between 25 and 50% reduction in fertilizer for maize. We can get, on our best trials in wheat, we can actually get it down to 85% reduction of fertilizer. But we don't get that consistently, and sometimes it can only be a few percent. So in wheat, it's that variability we need to reduce. In rice, it is incredibly economic for farmers, especially when you're getting yield increases of anything up to 30%, which we have managed. So you start looking at the socioeconomics of it, and the cost of the product is relatively low, but you still have to distribute it, it still has to be sold, and those have logistic costs and partner costs for your licensing companies. But even so, it's still all doable in commercial sense and in a public good sense as well. As I said before, the biggest cost is the cost of the packaging. And if I just add, we haven't yet worked out, and you might think this is something we should do, we haven't yet worked out what the benefit is of the mitigation cost for nitrous oxide. So there's interest in that, but you need to understand we are a commercial company. We're trying to get a product into the marketplace. There's all these additional benefits, and one of the reasons we talk to people like yourselves is in partnership that sort of work can be achieved, but it's not necessarily our role to actually carry that out.
Well, I have a question.
Yeah.
Maybe this is too early, but do you already have an idea what would be the cost of the product?
Yes, yeah. Well, going back to the gentleman over there, what we were looking to do was to basically give a substantial saving on the fertilizer cost for a farmer. So, and one of the difficulties is fertilizer costs go up and down. So, what the farmer wants is stability, and one of the things that we can offer is stability of yield in fluctuating fertilizer prices, because they can reduce the amount of fertilizer they have. So we've gauged everything according to how much fertilizers will cost in a given market. And so you then, essentially, we're giving a reduction to the farmer of about 40% on his or her nitrogen fertilizer use and costs. And you can work that back to any situation. But you're talking, this is really relatively low amounts. I'm not keen to actually give you figures because obviously we have competitors and we have to price accordingly to the right market. But we've done all the analysis, and it's a relatively low cost. All the price sensitivity analysis we did in the US came back as a major shock as to how readily farmers were prepared to purchase the product and gain, as much as anything, the sustainability benefits and nitrous oxide. We thought they would just want yield increases, but no, it's not just the case. Oh, yes, another question.
Another question from the comment.
Ah, damn.
For many poor farmers, smallholders in low middle-income countries that are under rain-fed conditions are facing high uncertainty. So if you have a drought, then you lose your crop. And you don't have, and many of those farmers do not have financial buffers. So very often in those circumstances, there is no levels of fertilizer used because of the risk of not recovering this cost. And if the situation is favorable, crops will get a top price. Now this is a product that is provided during
At seed, yeah.
So you're taking a huge risk at the onset of the growing season when you don't know if you're actually going to be able to recuperate those costs.
Yeah, it's a good point. And I think you'll find that the same issues actually apply in the developed world as well as the developing, although the failure of a crop probably wouldn't have such catastrophic consequences on life in the developed world. It's an interesting question because it is about how farmers view risk and what factors actually influence that. And farmers will use nitrogen fertilizers, as you say, to mitigate against risk. So we're approaching things in two ways. One is we're actually starting trials where we look to see where the benefits are accruing in time when you put on a top dressing. But we're also, more importantly, looking at a diagnostic that can tell you how many of the bacteria will actually be present in the crop, to give you the assurance that it will have an impact. And once we've got those two things working in tandem, then we feel confident it will give farmers the assurances they need. But ultimately, it will only ever come down to it being used so widely by so many farmers in so many circumstances that the farmers themselves know when to use it best. Is that a fair answer?
Yes, Simon.
Do you by chance brought a sample so we could try it out this summer?
No. It's an idea, but carrying milky fluids through the security people and the customs is never a wise idea. So no, on this occasion we hadn't brought any with us, but should collaboration evolve out of this visit and other visits, then yes, the product will be made available.
Okay.
Well, I'd just like to add that David and Nick will still be here tomorrow. They only have an appointment from 9 to 10, so if you'd like to meet with them after that, they're available.
Absolutely.
And well, with that, I think we close, and let me thank David and Nick for their presentation.
You're welcome. Thank you. Okay.